So I'm tasked with giving a brief research update. And I thought about what I'd like to say. And basically, it's going to be fairly general and conceptual. I thought that would be the best. So what you've been hearing from all the previous speakers is essentially the information and guidelines that have come from years and years of clinical research. Now, these guidelines and information have been the product of not just years, but also many different researchers, many different groups. And I think that every time I come to one of these symposia, I think it's really astounding what people are learning, what they have learned. It's tremendous. But I am a basic researcher, and what we focus on are trying to identify the key steps in disease progression. And what we think are the key steps are at the level of molecules and the level of cells. So clinical researchers use real human patients and gain knowledge from real human patients. And we look at a level much below that to try to identify new steps in disease progression. And we do this using a variety of approaches. I'm a biochemist, others are cell biologists, molecular biologists. We all come together and use various techniques. And the end goal is to use animal models in preclinical trials. Now, the goal for both of us, for clinical researchers and for basic research, is to improve the lives of people with our genetic disorders. And the way that we can do that is by organizing a well-developed clinical trial. And these clinical trials are the product of what we know from clinical research, what we've learned from basic research, and what looks to be a promising preclinical trial in animal models. So that's how it works. I'm going to talk to you a little bit about mouse models because recently a number of people, or at least over the last 10 years, a number of people have asked me these two questions. And I think that we should address them openly. So why did Losartan, oh God, what did I just do? I went backwards. So why did Losartan cure the Marfan mouse, but not people with Marfan syndrome? I've heard that question many times. And I've also heard people ask why we even use mouse models when mice are clearly not human. So one take-home message of our mouse models has been that maybe the things that we're finding are not relevant to humans because mice are not humans. So I'd like to talk to you a little bit about why we use mouse models. So there are two major advantages to using a mouse model. We can select mice that have an identical genetic background. So what this means is that people breeding mice for the last more than 50 years have bred strains of mice that are essentially genetically identical. We can't use humans that have this identical genetic background. The second advantage, which I think is really significant, is that if we can model disease in mice, we can look at when disease begins, when and how it progresses, and when and how it ends. If one only works in humans, there are certain practical limits to how humans can be studied. With mice, we can intervene or conduct experiments at any time during the lifespan of the mouse. So those are the two basic advantages. Now, I'd like to go over a little history. So these are the first two mouse models of Marfan syndrome. This one, MGR, was created by Lígia Pereira, who's now in Brazil, when she was a graduate student in Francesco Ramirez's lab. And this mouse model, C1039G, was created in Hal Dietz's lab. This mouse model was in a way kind of accidental. It was described in 1999. And what it shows is that reduced quantities of basically wild type or normal fibrillin-1 can lead to aortic disease. Now, this was something that we didn't know until we had this mouse model, that a simple reduction of fibrillin-1 underlies disease. And this can be normal fibrillin-1. So if you have 20% reduced, if you have fibrillin-1 that is reduced to only 20% levels, you could have Marfan syndrome. That's what we learned from that mouse. When Hal made this mouse, he modeled the mutation based on a mutation found in humans. And this mouse has aortic root aneurysm that grows. And it was debated for some time whether this mouse model was in fact the Marfan mouse and had, because it was more similar to human Marfan syndrome. This mouse, MGR, in fact dies from aortic rupture, but the rupture occurs in the ascending aorta. And in those early years, we used to debate whether or not there was superiority of one mouse over another. I would like to say that today, those kinds of debates miss the point. But let me continue. So it was in this mouse that antibodies to TGF-beta or losartan cured aortic aneurysm and a variety of other phenotypes, including fragmentation of the aortic elastic lamellae. Later, when the Ramirez lab looked at MGR with neutralizing antibodies to TGF-beta and with losartan, they found differential responses to both TGF-beta antibodies and to losartan. So, there. So I think that results from these mice may be confusing to you. And I know a lot of people have been confused, but I would like to argue that it is really important not to conclude that the results are not relevant because mice are not human. I mean, I think that has been something that's been out there, and I would like to address this issue. We are very fortunate in the Marfan world to have now multiple mouse models of Marfan syndrome. And I think that the correct way to look at these mouse models is not to ask questions like, is this mouse more a mouse model of Marfan syndrome than this other mouse? If you cure this mouse but not this mouse, does that say something? I think that those questions are not the most important questions. I think the most important questions are, what can we learn from these multiple mouse models that we're very fortunate to have. So on this slide, I've just summarized some of these mouse models. And this is a spectrum of disease severity. This is for aortic disease for these various mouse models. So at this end, there's wild type. This mouse has a normal aorta and a normal aortic wall with its nice elastic lamellae. On this end is the complete fibrillin-1 null. So this mouse has no fibrillin-1, and it dies 2 weeks after birth from aortic rupture. This mouse, MGR, which is the one that I told you about, has 20% the levels of normal fibrillin-1, and it develops aortic root aneurysm as well as dissection and rupture. Now, this mouse model, C1039G, I also told you about. It never does this. It never dissects and ruptures, but it does develop an aortic root aneurysm. We also have a mouse model that shows aneurysm without rupture, and we call that model GT8. And we have a new mouse model that we call H1 delta that develops no aneurysms, but it shows histological findings indicative of fragmentation of the elastic lamellae, and we call this a mouse model of microdissection. So when we take this allele of fibrillin-1 and this allele of fibrillin-1 and make a compound mouse, now we have a mouse that does die usually around 5 months from aortic aneurysm and dissection. So here's my theoretical slide. So you can have a genetic cause of a disease. It can be a mutation in FBN1, a mutation in COL3A1, a mutation in TGF-beta receptor genes. And what we want to know is how can we block the progression of disease? How can we ameliorate the progression of disease? And what we know, this is just biology. What we know is that this genetic cause will lead to multiple different pathways. And way down here, this could be perhaps the TGF-beta signaling pathway, or this one up here could be the nitric oxide signaling pathway, or in the case of COL3A1, there could be effects on the protein kinase C pathway. And so if one blocks these pathways, so for example, if you block TGF-beta signaling in FBN1, I think we now know that we get a variety of different effects, and we do not get a cure of Marfan syndrome. And the reason for that is that it's way downstream from up here. And when we block things down here, they can, biology has a way to move things in another direction. So all of these pathways relate to each other, and we have no idea really how to connect all these different pathways. But we have different approaches to try. So what we're trying in my lab is to simplify all those arrows by using mouse models. So we have a mouse model that shows only microdissection, no aneurysm. We have a mouse model that shows aneurysm, no rupture. And we have a mouse model that shows aneurysm with rupture. And by analyzing these three different mouse models, we feel that we can simplify these pathways that go to the different mouse models and show how they interact, show more directly how these pathways interact. And this is important because any type of therapeutic, we want to know if we block here, does this in fact block the formation of microdissections? If we block something here, does this in fact inhibit the pathway to rupture? The same as here, 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 and here. So what we think is an important approach is to be able to organize these pathways in a reasonable manner around specific phenotypes, and that will go a long way towards simplifying that this crazy slide. This is the state of knowledge now. And with mouse models, I hope that the state of knowledge in the future will be more like this. So in the previous talks, you heard a lot about outcomes. I think it was Luch who talked about not knowing what the outcome measures are for exercise. So this is what we researchers work on. We work on defining what the outcome measures should be. And I think that it is better to focus on outcome measures specifically, rather than on this general concept of a cure. I think that would be very helpful in terms of interpreting research. Right now, as you've heard, we look primarily at aortic size, at the aortic rate of growth. We also use a lot of histology. In humans, this is from removed tissues. And in mice, we can do histological analyses quite easily. But even then, there's a need for better imaging from clinical researchers for dissections, better scanning procedures, and this will happen in the future, I think. Things are coming down the pike that will lead to better imaging methods. From the basic research point of view, what we would like to do is to identify those specific molecular markers and those specific cellular markers that play important roles in the disease progression. And it's very important to researchers not just to identify these specific markers, but to be able to quantitate these specific markers. And that is because if we want to measure an outcome in one of these markers, an outcome as a result of a drug therapy, for example, these measurements have to be quantitative. We can't just look at, for example, a histological slide and say, well, that slide looks like it has no PSMAD immunolocalization, whereas the disease slide does have PSMAD immunolocalization. We feel that we need much more rigorous quantitation of these markers. And another thing that this could lead to in the future are plasma biomarkers. And these plasma biomarkers, I think some of you know that I've been working on fibrillin fragments as plasma biomarkers for monitoring disease. I think that those studies are still really good and really potentially useful. But these studies could also lead to similar types of plasma biomarkers that could be useful to monitor the status of disease in a person. So if you know that you're at risk for dissection, if you know you have an aneurysm, I think it would be pretty great if someday, like a diabetic, you had an in-home test and you could just prick your finger and see that your plasma biomarkers are stable and go about your day. I think that that's one of the hopes that we have for the future.